Good day, everyone. My name is Michael Akitiboso. I'm a mentor at OFIS, a first-year doctoral student here at the University of Alabama in the Department of Biological Sciences. Today, I'm going to be telling you what my research is all about and how we carry out our research. So basically, in my lab, we work on microbial natural products. Microbial natural products are specialized metabolites that are not necessarily involved in the survival of the organisms, but have found vast application in medicine. And these natural products are usually found in gene clusters, in what we call biosynthetic gene clusters. Now, due to the advances in genome mining and sequencing and all that, we have thousands of unannotated biosynthetic gene clusters, which means that there are thousands of drugs that we haven't even discovered. So what my lab will try to do is to use bioinformatics tools to predict these different biosynthetic gene clusters that can possibly produce novel glycosylated natural products. And secondly, we are going to try to understand the different reactions that their biosynthetic enzymes catalyze. In order to do this, we'll use a lot of bioinformatic tools, biochemical reactions, and structural biology. So I'm going to be showing you around my lab, and we believe that by doing this, we'll be able to produce natural products that kind of solves the problem of antimicrobial resistance. So basically, this is my lab space, and this is like how a normal lab looks like, a more biochemical lab looks like. I'm going to say this first is what we call our sonicator. So remember I told you we do a lot of biochemical reactions. This is basically used to sonicate our bacterial cells. By sonication, I mean breaking down our cells to release the protein. This is what we call the incubator. As you can see, presently I'm currently producing, overexpressing a particular protein. So our protein is basically going to be released into the cell, into the media, and then we carry out further process. So this is a protein production in progress. So this is my workbench where I carry out like a lot of my biochemical reactions to understand the biosynthetic pathways of our enzyme. And this is a centrifuge. Basically, after our protein production and after lysing ourselves using the sonicator I earlier showed you, we are going to centrifuge to divide the cells from the protein produced. This is basically what we call the electrophoretic machine, and what it does is to allow foreign DNA to enter into our cells. So basically, we are kind of pulsing our cell using high voltage to allow foreign DNA to enter. And this is the thermal cycle, what we know as the which carries out the polymerase chain reaction, and it's used to produce thousands of copies of a target gene. And this is also a PCR. This is what we call the incubator, and basically what it does is to grow our organism. For example, I work on an organism called the Streptococcus, Streptomyces hygroscopicus, and it usually grows at 30 degrees Celsius. So that is why you have the temperature set here at 30 degrees Celsius. This is our size exclusion chromatography, and basically we use it to achieve purification of our protein. So if our protein is present, we are going to see a sharp peak here. This is also a size exclusion chromatography machine. The difference is just in the column. So it depends on the size of our protein. It helps us to know the column to use. And like I said, we do a lot of bioinformatic analysis, and this is one of my colleagues working on the laptop. So basically, these laptops, we use it for bioinformatic analysis to identify our biosynthetic gene clusters. By combining all these tools together, we believe we'll be able to predict novel glycosylated natural products which solves the problem of antimicrobial resistance.